like to read the preamble of the Constitution. We, the people of India, having solemnly resolved to constitute India into a sovereign, socialist, secular, democratic republic, and to secure to all its citizens justice, social, economic, and political, liberty of thought, expression, belief, faith, and worship, equality of status and of opportunity, and to promote among them all fraternity, assuring the dignity of the individual and the unity and integrity of the nation. In our Constituent Assembly, this 26th day of November 1949, do hereby adopt, enact, and give to ourselves this Constitution. Now, I would like to hand over the room to our moderator, Professor Dr. Srinivas.
Thank you, James. Thank you. Telangana Jana Vedika Zoom online meeting ku చేసినటువంటి ప్రొఫెసర్ పద్దయ్య గారికి, వేదిక సభ్యులు, పెద్దలు, మిత్రులు అందరికీ సాయంకాల నమస్కారాలు తెలియజేస్తూ అందరికీ స్వాగతం, సుస్వాగతం. తెలంగాణ జన వేదిక ఏర్పడ్డ దగ్గర నుండి ఇప్పటి వరకు నేటి వరకు 105 కార్యక్రమాలు, అంటే 405 సమావేశాలు ఏర్పాటు చేసుకున్నాం. కానీ ఇవాళ అంశము చాలా కొత్త అంశం మనకు. నియానికి కూడా వేదికలో చాలా మందికి మానవ జన్యు శాస్త్రం అనేటువంటి విషయం గురించి చాలా మేరకు తెలియదు. సో మానవ జన్యు శాస్త్రము ఇటీవల పురోగతులు అనేటువంటి అంశము మనకు కొత్త అంశం అయినప్పటికీ ఈ అంశం గురించి మనము తెలుసుకోవాల్సినటువంటి అవసరం ఉంది. ముఖ్యంగా సోషల్ సైన్స్ చదువుకున్న వాళ్ళు కానీ, న్యాచురల్ సైన్సెస్ కానీ, ఆర్ట్స్ చదువుకున్నటువంటి వాళ్ళు యొక్క మానవ జన్యు శాస్త్రము అనే దాని మీద మనము తెలుసుకోవాల్సినటువంటి అవసరము, బాధ్యత కూడా ఉంది అని చెప్పేసి నేను భావిస్తూ, మానవ జన్యు శాస్త్రము ఇటీవల పురోగతులు అనేటువంటి అంశం అయిన ప్రొఫెసర్ పద్దయ్య గారు, వారు డిపార్ట్మెంట్ ఆఫ్ హ్యూమన్ జెనెటిక్స్, ఆంధ్ర యూనివర్సిటీ విశాఖపట్నం నందు వారు పనిచేశారు. వారు హెడ్ డిపార్ట్మెంట్ గాను, బోర్డ్ ఆఫ్ స్టడీ చైర్పర్సన్ గాను, మరియు డీన్ ఫ్యాకల్టీ గాను వారు పనిచేశారు. వారు దగ్గర అనేక మంది పిహెచ్డి స్కాలర్లు, ఎంఫిల్ స్కాలర్లు, అంతేకాకుండా ప్రొఫెసర్ పద్దయ్య గారు వారు దేశ విదేశాల లోపల అనేక కాన్ఫరెన్స్లు అటెండ్ కావడం, అదేవిధంగా వారి యొక్క రీసెర్చ్ పేపర్లు కూడా జాతీయంగాను అంతర్జాతీయంగాను పబ్లిష్ కావడం కూడా ఒక గొప్ప విషయంగానే మనం భావించాలి. అందులో వాళ్ళు సుదీర్ఘంగా 30 సంవత్సరాల కాలం పాటు ఈ యొక్క అధ్యాపక వృత్తిలో ఉండి అనేక మంది విద్యార్థులను తయారు చేసేసి వారి దగ్గర పనిచేసినటువంటి వారు దేశ విదేశాల లోపల అనేకమైనటువంటి పదవుల్లో వారు కొనసాగించడం కూడా మనము ఈ సందర్భంగా గుర్తు చేసుకుంటూ, ప్రొఫెసర్ పద్దయ్య గారు మానవ జన్యు శాస్త్రం ఇటీవల పురోగతులు అనేటువంటి అంశం మీద వారిని మాట్లాడవలసిందిగా సాదరంగా ఆహ్వానిస్తున్నాను.
Right. Thank you very much, sir. And in fact, it's my so am I audible, sir?
Yes, sir. Please. Yeah.
Right. So thank you very much for the Telangana Jana Vedika for inviting me to give a lecture on the recent trends on developments in human genetics. I'll try first of all, I'll just want to give a comprehensive idea of how hereditary things will be passed on from one generation to the other. Basically, heredity means how the characters are being passed on from parents to offspring, from one generation to the other, and it keeps on going. In doing so, there are some principles, and according to those principles only the hereditary material gets passed on to the next generation. So I'll start with this particular so when you actually go before the scientific this thing, Hippocrates, the father of medicine, actually told that the semen will be secreted from each and every part of the body, including the soft parts and also the hard parts. The basis is, if it is not produced by the entire body, how it actually gets this thing. So how bald people are getting bald people, squint eye people are getting squint eye, then the tall people are getting tall people, all these things, how it is possible if it is not there, if it is not passed on from each and every part of the body. And later, there are different things. So whatever characters we have are already there in a miniature form in the semen. If it is not there, how actually they get developed? That was the thinking. And later, Aristotle also gave us some this thing. Aristotle, he said, if the male impetus is more, then we'll get sons. On the other hand, if the female impetus is more, we'll get daughters. And daughters look like their mothers and sons look like their fathers. But all these things are in the absence of concrete scientific evidence. And then Plato, we all actually blame Hitler for eugenics and all that thing. But Plato himself started this kind of eugenics. He said, the best of men should marry the best of women, and the children of these couples should be the responsibility of the government. This is no, it's fine. But the problem with Plato's thing is, on the other hand, the children of the inferior have to be dispensed with. That is the problem. And because later science proved that even the inferior people can have genius children. That is why in that sense, Plato was, I mean, you can find fault with Plato. But then again, there was later whom was actually discovered. Then the emphasis has shifted to the ovum. Earlier, people used to think that everything is in the miniature form in the semen, but that thing got shifted to ovum. So the ovum controversy that the ovum contains all these things. Sometimes the sperm contains all this, those things. Later, during this 1772, that this thing, Mendel, who is considered as the father of genetics, did some epoch-making experiments with garden peas. But even before Mendel, Gartner and other people did similar experiments, but all of them failed basically because they wanted to take all the characters into consideration. Mendel was successful basically because he took one character at a time and then discovered how it is possible, how it is developing generation after generation. That was the great thing. And then Galton actually, again, during that time, he also made some experiments, Sir Francis Galton. These two people laid a very concrete foundation for the study of genetics, and later it's then all history. When we actually go, whatever we are at now, at one point of time, we are all are like this single cell. This single cell contains all the genetic endowment that is essential and necessary to develop into a full-fledged individual later. So what are those ingredients that are embedded in this cell? This is what is called zygote, and this zygote is formed upon the fusion. This cell is the basic functional unit in any living organism. To give some kind of an idea, human beings have 100 trillion cells. Each one of the cell contains exactly the same genetic endowment as that is there in this cell. And how this cell, what is called the zygote, is formed, I'll explain. In males, spermatozoa are formed. And this spermatozoa, there are two kinds of spermatozoa. 50% of the spermatozoa, right? To give you some kind of an idea, human beings have 46 chromosomes. And these 46 chromosomes, out of these 46 chromosomes, 44 chromosomes are common both for males and females. So you cannot say out of these 44 chromosomes, which chromosome is a male chromosome, which chromosome is a female chromosome. To that extent, that means each and every character of the human being, or for that matter, any living organism, has a genetic basis. If that is true, 46 out of 44 out of 46 chromosomes are exactly same between males and females. You cannot say that the males are superior to the females. In fact, if at all any superiority is to be there, that is with the females, because out of these two chromosomes, X and X, in addition to these 44 chromosomes, are there in females, whereas in males, X and Y. This Y chromosome, father to son. This inheritance is from father to son, whereas females get one X chromosome from the father and another X chromosome from the mother. And these two X chromosomes are bigger than the Y. Is a very teeny chromosome, and it does not have a lot of chromosomes except, a lot of genes except the male determining factors, testis determining factors, and all those things. So when you take the genetic component as such, females are better endowed when compared to the males. And in the spermatozoa, there are two kinds of spermatozoa. 50% of them are carrying, I told you, these 44 chromosomes are known as ordinary chromosomes. And then these two sex chromosomes, X and Y, are known as sex chromosomes. So the X and Y chromosomes pair in the division of cells. And 50% of the spermatozoa contains X-bearing spermatozoa, and another 50% Y. On the other hand, the eggs are always 23X. So the reduction of these 46 chromosomes to 23 is an essential step to maintain the fidelity of the human heredity, because we inherit all our 50% of our chromosomes from the father, 50% from the mother. And then when we actually produce the spermatozoa at the end, the genes that are there within this 23X, 23Y get shuffled. So every time an X chromosome, a spermatozoa is produced, the genes get shuffled. So is the case in here. The egg also, every time an egg is produced, the genes are different. So here I'll just spend 2 minutes in explaining these things, because this is a very important thing. In the entire reproductive life of a female, the around 450 to 500 eggs are produced. And these eggs are produced even when the female was in her mother's womb. And they are embedded in the in their eggs, this thing. And they are there taking rest when the female is born, and when it is growing around 11-12 years, out of the stock of 450, one egg completes the first meiotic division, and then the egg is produced. If that is fertilized, then again that develops into an individual. On the other hand, the spermatogenesis process per se starts producing spermatozoa when the male becomes an adult. So when the same egg is fertilized by a Y-bearing spermatozoa, you will have 46XY and it develops into a male. On the other hand, if the same egg is fertilized by an X-bearing spermatozoa, it develops into a female, 46XX. So in our society, in many of the societies, people blame that for not getting sons. The mother is blamed. If at all something is to be blamed, the father is to be blamed because he produces two kinds of spermatozoa. Even that is there inherent because he doesn't want to do this thing. But one thing now, especially you must have observed that when people go for in vitro fertilization, there is overwhelming majority of the children will be males, because what they do is they actually spin the spermatozoa. When they actually spin the spermatozoa, because the Y-bearing Y chromosome is a smaller one, it comes to the top. When that spermatozoa, when actually the doctor uses that particular spermatozoa to fertilize, it mostly overwhelmingly contains Y-bearing spermatozoa, which results in the sons. Right. When once this zygote is formed, it divides, and in 9 months it develops into a newborn baby. And in around 18 years, that will become 100 trillion cells. These 100 trillion cells are in the form of 200 different cell lines and the whole of the human body, organs and all that thing. So most important thing is, out of this 100 trillion cells, each cell is connected through the genes. The functioning of these genes, again, I'll explain it in detail later, but for the time being, when you take the genes after the discovery of the Human Genome Project, earlier people used to think that there are 100,000 genes that are to be there in the human being. But later on the publication of this Human Genome Project, they realized that we don't need to have 100,000 genes. Why they actually came to that part of conclusion for to have 100,000 genes is thinking that when you take the human body, there are 100,000 different types of proteins and enzymes. Beadle and Totem, two scientists who got a Nobel Prize, experimentally demonstrated that you have to have one gene for the production of a protein and the enzyme or an enzyme. When the whole human body has 100,000 different kinds of proteins and enzymes, scientists thought that unless we have 100,000 genes, there is no way for the production of 100,000 proteins or the enzymes. But after the publication of the Human Genome Project results, then the reassessed that we don't really need 100,000 genes. It does not mean that the Beadle and Totem were wrong, because they experimentally demonstrated that you have to have one gene for the production of an enzyme. They are not wrong. But here, what happens is, one gene produces one protein, another gene produces another protein, another gene produces. This gene in combination with this gene can produce yet 2-3 genes. When you take these things into consideration, we don't really need 100,000 genes. 25,000 genes are eminently more than enough to explain all the complexity that is there in the human body, right? So next we will go for the next thing. So this is the human. I told you the cell is the basic functional unit. And within this cell, we have this 46 chromosomes in human being and around 25-30,000 genes. And the most important thing is, even these 30,000 genes are basically made from small components known as, I'll explain, small particles. What are those particles? I'll explain here. Okay, this is the chromosome and which can be stretched. Actually, when you stretch this DNA from each cell, it will be.
Do you want to share the screen, Sid?
Sir?
Do you want to share the screen?
Yes, sir. You are not seeing the screen, sir?
No, we have.
Okay.
Yeah, we
So,
are unable to see, sir.
You are unable to see? Oh, sorry. Yeah. Okay.
You have to share. You have to share it, sir.
Right, sir. Okay. I'm going. మీరు మరి ఇందాక నుంచి ఒక మాట చెప్పలేదు సార్ ఇది ఓకే I'm actually finding it little okay so share okay I'm sorry.
Yes, now we can see.
I'm sorry. I'll just kick. I'm very sorry. I mean, you should have. Okay. All right, sir. Then, right. So I'm talking about. Okay. Because there is no point. Right. I'll show at one point of time. This is the thing. Here, I just want to emphasize. In each ejaculation, there will be 300 million spermatozoa. Each spermatozoa is unique in its construction. So, when in intercourse, when 300 million spermatozoa are deposited, there is great competition among these spermatozoa to reach the egg. Egg is actually released into the fallopian tubes, and all the spermatozoa are deposited in the vaginal canal, and they have to go some distance to reach the egg, and all these spermatozoa are actually trying to reach. So when once a particular spermatozoa enters the egg, then the egg will not allow any other spermatozoa to come, because that is the most important aspect, right? Then immediately, why the spermatozoa, I mean, the egg is 80,000 times bigger than the spermatozoa. The spermatozoa is the smallest cell in the human body. On the other hand, egg is the biggest cell in the human body. Why it has to be so big, 80,000 times bigger than when compared to the spermatozoa, is basically because when once the fertilization process per se takes place, the division starts immediately. Every 24 hours, one cell will become two cells, and it goes in an exponential way. For the cell to divide into two, it needs basic ingredients. All those ingredients are there in a reserved material, as a reserved material in the egg. That is why it is 80,000 times bigger, till this zygote actually travels and implants itself in the uterus. When once it attaches itself to the uterus, it gets all the ingredients from the mother's blood circulation, oxygen and other things. Till that time, it has to sustain. It takes a week. So in that period, many cells actually have to be produced. For them to be produced, it has to have all the reserved material. Right. I'll just, because right, I'll go quickly. Right. From the current slide. Okay. Then, all right. These are the things. Right. So the most important thing is, this is the entire human being cell, chromosomes, 30,000, and these things are formed from smaller things. Which are those smaller things? I'll tell. Right. This is the chromosome. When it is stretched, I told you, I was telling that it will be 6 feet long, each cell. Like that, there are 100 trillion cells. Each one is derived from the same thing. How exactly the fidelity of these genes will be maintained, I'll explain in a minute. So these are the different ways of actually doing these things. Oh, okay, there is some. Oh, okay. Sorry. Just 1 second. There is some block. Close the program. No. Wait for the program to respond. Sorry.
Sir, you can click on Alt tab, sir, if you want to.
Alt tab. Okay.
Alt tab at the same time, sir.
Right. Revocation for certificate for this. Okay. It's available. Do you want to proceed? Yes. No. Okay. No. Sorry. Alt tab. Okay. I'm sorry.
Sir, do you want to exit?
Yeah. I got it, sir. Right. Okay. So, all right. So from this side. All right. Okay. Share. Okay. Malla idu poinda. Right. Okay.
It is visible, sir.
It's visible. Okay. All right.
Yes.
Right. So when you take these chromosomes, these are the 46 chromosomes. And now we can check at chromosomal level whether there is any difference as far as the chromosomes are concerned. That, and all the chromosomal component has to be there. If any of these chromosomes are missing or added, or even a part of the chromosome is missing, that can lead to gross abnormalities, right? And then, so these are the things, I mean, at a different, let us, this thing, right? The whole Mendelian genetics was started in 1900. In 1940, the microbial genetics, because basically whether it is microbe or human, the basic construction of the genes are from the same building blocks. Because the microbes, especially bacteria, virus, and all that thing, are smaller organisms, and their lifespan is so small, you can start it when scientists took those things because they can do a lot more experiments in days, weeks, and all that thing. If you want to do the same experiments with human beings, the scientist's life and human life is the same. That is why scientists focused more on microbial genetics. And in 1970s, the gene manipulation, the new genetics has started, and how that those things, because right, and these gene manipulations, molecular genetics and microbial genetics enormously helped human genetics in understanding how the genes function while being passed on from one generation to the other generation. But for the contribution of this microbial genetics and molecular genetics, things would not have been as clear now as if their contributions were not there. Then, right, these are the things, the normal spermatogenesis from each spermatozoa. The spermatozoa are produced only in testes. And from each testis, for each cell, 4 spermatozoa are produced. On the other hand, from each oogonia, only 1 egg is produced. I told you sometime back, in the entire reproductive period of a woman, there will be no more than 450 eggs. And these eggs start getting discharged when the first menstruation, what is known as menarche. From then onwards, every 28 days, 1 egg will be released. If it is fertilized, then the release of the eggs will be stopped till the delivery of the child, and also after delivery, postpartum amenorrhea for few months, and later it gets resumed. When all the eggs that are there embedded in the ovaries are exhausted, the women reaches menopause. Afterwards, they cannot have children, right? So from each this thing, I told you that the egg is 80,000 times bigger than the spermatozoa, and that is for the reasons, right? Then, right, these are the different things, right? 22 pairs of chromosomes, which are same both for males and females. And the only difference is XY in males, XX in females. As far as these 44 chromosomes, autosomes are concerned, you cannot say whether it is a male chromosome or a female chromosome. And I told you, there is a gene behind each and every character. If that you take, you cannot say that females are inferior or males are superior, all these things. If at all, I told you that the females are superior because see, for the simple reason, there are many other reasons why females are more genetically endowed, better endowed, strong, and all that thing. Apart from these things, the very process of development of the child, egg into a child, is given naturally to the woman. That means they need a lot more genetic machinery to do this thing, which is not there with males. For that simple reason, women are superior to men. Of course, there are many other things to prove that women are superior, right? Right. These are the things. Then, right. So when there are some problems with the chromosomes, what kind of things will go quickly? In the process of the distribution of genes, I mean, there is sometimes what is called non-disjunction. Because of this non-disjunction, whatever, we actually received all our 46 chromosomes, 43 from my father, 43 from the father, and these things paired. And then the genes get shuffled every time a spermatozoa is formed. To give you some kind of this thing, when you actually pack of cards, every time they get shuffled and divided into two, like that, the genes will be distributed to each spermatozoa, each egg in that way. That is why each egg is unique in its genetic constitution. Each spermatozoa is again unique in its constitution. We do not know which spermatozoa out of this 3,000 spermatozoa fertilizes the egg. And out of this stock of 450 eggs, which egg released at what time? And out of this 450, maximum earlier used to have 10 children or this thing. So out of 450, only 10 eggs developed into children. Now it is 1 or none or maximum 2. Out of 450, which egg is released what time, and that particular egg is fertilized by which spermatozoa, that determines the qualities of the children, right? And then in the process, right, suppose this is a Turner syndrome where one of the chromosome is missing, one of the X chromosomes is missing, and she can't, when though apparently is a female, she is not a perfect female, she cannot have children afterwards. Similarly, if something is given, it's a syndrome where extra X chromosome is there, 47 XXY. Males have to have only 46 XY. When, because of non-disjunction of these chromosomes, one extra X chromosome is added, he is apparently a male, but he can't be a female. Similarly, here Down syndrome, an extra 21 chromosome is added. Everything is perfect, but an extra chromosome addition actually makes these people, you must have observed some of these people, they are mild and intellectually not this thing. And that is basically because an extra chromosome is added. And similarly, so another kind of extra 21 chromosome, trisomy, we have to have every chromosome in twice. One we got from the father, one from the mother. When other than these two, if something is added, and how that actually, there is a mechanism how this actually happens, but that is normal, not normal process. It's an abnormal deviation from the normal process of fertilization and all that thing. And then, so there is a familial Down syndrome. You must have observed that generally this happening of Down syndrome happens in families, right? And there are some reasons for that. Let us talk this thing, right? And earlier, they used to get this Down syndrome when women actually got these children when they are more than 40 years and all that thing. So the idea, they explained that because these eggs are formed and embedded in the ovaries of the female when she was in her mother's womb, when she actually conceives, that means that particular egg is in a resting stage for all the 40 years. Because of it is in a resting stage for all the 40 years, the chromosomal distribution was not perfect, and that actually is leading to this kind of a problem. That was the thing. But later they found that this need not necessarily be from the mother. It can also be from the father, because spermatozoa is being produced afresh. It takes from primary spermatocyte to the spermatozoa. The production is only 2 to 3 months. But even then, this is happening. Now we have a technology to identify where this extra chromosome comes from, whether it comes from the mother or from the father. Earlier, this explanation was it is coming from the mother because 40 years of resting stage, this is happening. But now, even without resting, also it is coming from the father. Later, right, there is this kind of this thing. Let us go, right. It's a Down syndrome, different. It could be male or female, because this extra 21 chromosome is an autosome. Autosome in the sense, it has nothing to do with the sex chromosomes. Right. So here another important thing is, we don't need, the entire chromosome need not be added. Even when a piece of the chromosome is missing, that can lead to some kind of a problem. Here in Edwards syndrome, on chromosome 5, a small piece is missing, that is actually causing some problem. Similarly, there is another this thing in 13 chromosome, a small piece is missing, that can. So the thing is, this genetic endowment has to be distributed in a perfect way. Anything other than this perfect distribution of the genetic material can lead to a lot of problems. It could be this thing, right? In chromosome 5, a small thing is missing. It can lead to this thing kind of, it's a card. These people wailing resembles that of a cat. That is why it is Cry-do-chart syndrome, right? There is, again, not only all the chromosomes have to be there, they have to be in a perfect condition. If some of these X chromosomes are fragile, that means the DNA that is there on these things is not intact. That can also lead to some kind of problems. What kind of problems? Right. So this is what is called earlier thalidomide for morning sickness. Earlier women used to take some kind of drug. The very use of that drug led to the birth of different people with different abnormalities. But of course, that is now completely banned. But another important thing is the distribution of genetic material has to be perfect in all animals. But in plants, in some of the plants, an extra set of chromosomes can be more beneficial. And especially see in cotton, tetraploids, pentaploids, they actually give better yield. But this kind of a thing is detrimental in animals. Right? Okay, let us go for these things. Right. Right. So in 1990, a consortium has started the Human Genome Project. The idea is to identify all the basic ingredients of these things. Let me explain this particular DNA structure, because this is the crux of the whole genetics. See, I told you that if you take the DNA in each cell and stretch it, it will be 6 feet. And that 6 feet contains 3.2 billion base pairs. These are base pairs, right? So even here, there are only 4. There are 4 nitrogenous bases: adenine, thymine, cytosine, guanine. These 4 are the most important thing, because the pair, again, the pairing is very unique. Adenine pairs only with thymine, cytosine pairs only with guanine. And these pairs are in each cell 3.2 billion bases. The rest is attached to a sugar molecule that is attached again to phosphate deoxyribose phosphate molecule. The phosphate molecule is same, the sugar molecule is same. The only difference is the sequence of this adenine and thymine and these nitrogenous bases. Why the sequence of these things are so important, I'll explain in the next slide. Right? So because I told you that these are right. Within this thing, 3.2, one small piece is there that forms into a gene. For example, I'm dark. That means in my skin cells, the genes produce more amount of melanin pigment. The type of genes that are there in every individual which produces melanin is same. But the quantity of melanin pigment produced determines the skin color. If somebody that is very fair, it's the same gene, but it produces less amount of melanin pigment. So it holds good for each and every gene, right? Then why the sequence of that particular thing is so important is basically because these nitrogenous bases, 3 taken at a time, produces 1 amino acid. The genes produce proteins or enzymes. The basic building blocks of proteins or enzymes are amino acids. So the sequence of these amino acids determine the functional capability of the protein or the enzyme. And the sequence depends on the sequence that is there within this gene. And within this gene, 3 taken at a time actually forms a codon, and that codes for 1 single amino acid. When the code changes, the amino acid sequence changes. When the amino acid sequence changes, it gives, I'll just give one example. For example, all of us are perfectly normal. That means in our red cells, the hemoglobin production is perfect. In some individuals, when a small change within this hemoglobin gene can lead to some kind of hemoglobin, which will lead to sickle cell trait. If this happens in both these strands, those people die of sickle cell anemia. So the entire genome is perfect. One single mistake in one of the genes can cause havoc. So in that sense, this sequence is so important. And right here, I request all of you to focus this thing. So when we take this thing, I was talking about this 6 feet DNA, and within this 6 feet DNA, 75% of the DNA sequence is present only once. And out of that 75%, less than 10% sequence actually constitute the functioning genome. So what the other more than 70% of the genome is doing, still not very clear. So a single copy of this DNA, less than 10% actually codes for proteins and the enzymes. What the other things? Another 25% out of this, 15% is in the form of signs and lines. And again, I think you must have heard about the DNA fingerprinting. DNA fingerprinting is based on this particular satellite DNA, which is present in 10%. And this DNA sequence is the same repeats. And these repeats can be, and the repeats in each and every individual is unique. And now the scientists can identify, because this DNA fingerprinting is to identify thugs, rapes, questioned paternity, and all that thing. These thieves are becoming so smart that they are not leaving any fingerprints. But in the scuffle, there is every possibility for them to keep some of their biological material, like some hair, some scratch of the body and all that thing, from which DNA can be made. And that DNA, from that, this profile, profile can be made. And this profile, the police will go after all the suspected people. They get blood samples from that, and then the DNA is made. And that DNA is also studied for this DNA fingerprinting. This profile matches only with that kind of a person who actually committed the crime. It will not match with any other individual. Suppose the police have found some suspected and the actual culprit was not there, then it will not match. It's so, that is why it is called fingerprint, because we all know each fingerprint is so unique. Similarly, from the DNA, we can also identify the pattern, which is unique. From that, you can actually identify. But our important, the geneticist focus is on this particular DNA, which is present only once. And that once, even with that, only 10% is actually constitute the genes. And if there are any mistakes in that particular thing, that causes havoc. All right, then see, whether it is yeast, fruit fly, roundworm, mustard, human, the basic building blocks from which all individuals, including plants and all that thing, were made is basically this thing. When you take the entire genome of yeast, it is 12.1 megabases, means million base pairs. When you take the fruit fly, when you take the roundworm, this thing, when it comes to human beings, it is 3.2 billion bases. That actually, this particular genetic material make human beings, this particular genetic material make this plant, this into a roundworm, fruit fly, yeast. Even from bacteria, virus, the basic building blocks are exactly the same. And that is the most important for all genetic engineering experiments. This whole of genetic engineering experiments are possible only because the basic building blocks of all organisms, starting from virus, bacteria, plants, animals, human beings, are basically made from these things. You will understand, right? Another important, I'll show another this thing. From this thing, you'll write. The sequence of this DNA is so important because I told you we have 3.2 billion bases, and some differences within this sequence will make all the difference. See, for Bush, this is this sequence, and for Obama, for Laden, for Sonia Gandhi, for this thing, every individual out of these changes are from this 3.2 billion bases. When we know the 3.2 billion bases, we can tell which individual has what kind of strengths and weaknesses. Now we have the technology to identify the basic human genome sequence. Again, within this genome sequence, there are two kinds. I was telling that only less than 10% of that sequence which is present only once actually constitutes the genome, that is called as exome. Any changes in that exome can cause lot of problems. On the other hand, if there are some changes within this DNA sequence other than these coding regions, it may not cause great problem. So is the case in those 25%, 15%, even if there are big changes, there is no problem. But if even small changes are there within that less than 10% of that 75% sequence which is present only once, that causes all the problem. So even in this 3.2 billion bases, every 2 individuals on this globe is common to the tune of 99.8%. But that 0.2% changes within this genome sequence changes, that causes all the difference. Because to emphasize this particular point, I'll give one example. I was telling that normal hemoglobin, all of us, we got normal hemoglobin, we are perfectly normal. And some unfortunate people where one nitrogenous base is replaced by another nitrogenous base can cause sickle cell trait, in the sense when the oxygen tension is reduced, their red cells will become sickle shaped. When they become sickle shaped, they cannot pass through small capillaries. When they actually are passing through small capillaries, because they'll become sickle shaped, because generally the red cells are round, and when they are round they can pass freely. When they become sickle shaped, there is every possibility for them to get stuck. When they get stuck, our immune system thinks that something untoward is happening and then phagocyte those things, and there is every possibility for that individual to die if that particular hypoxic condition continues for longer time. In some other individual where this kind of mistake happens in both the genes, they will die of anemia in adulthood. So that is the thing. In each gene, these kind of changes happen. All of us are normal, means nothing untoward has happened in those genes, which is very important, right? So then now we have the technology, right? Another very important thing, right? Then I'll go further. So how the gene actually, because this is too technical, unless you have some biology background, it's very difficult to understand. But nonetheless, I'll just give another. This is an example. So the genes, the way they function in bacteria is very similar this way they function in human beings. I'll explain. For example, bacteria is growing. The conditions are favorable. Then the X, Y, A, these genes, they produce all the things that are needed for the bacteria to flourish. So when the circumstances become difficult, then what happens? The operator, there is actually repressor protein is produced. It actually blocks the functioning of these genes. So it shuts itself, protects itself. When the again environment become favorable, then it becomes, see, another mRNA is produced so that the repressor protein is taken away. Then these genes actually function and it flourishes. That kind of mechanism is there in human beings. I'm talking about one gene, how it actually works. This is the same thing for the proper functioning of all the 25,000 genes that are there. Here, another important point has to be emphasized. Though we have, I told you, 100 trillion cells, and these 100 trillion cells are organized into 200 cell lines. And these two cell lines, for example, all of them contain all the entire genome, but all the genes are not functional. They don't need to be functional in each and every cell. It all depends on the functional activity of this, of the cell, of the tissue, of the organ. To give you some this thing, for example, the very functioning of the brain is different. The set of genes that are functional in the brain genes are entirely different from the set of genes that are functional from heart, from liver, from kidney, from skin, from lungs, in each and every organ. So that kind of which set of genes are to be active, that again there is another set of genes which will oversee which set of genes have to be functional in this thing. That is why I explained this particular thing in a bacterium. In a bacterium, if this situation is favorable, all the genes function and they flourish, they divide, they proliferate, they grow in numbers. On the other hand, if the environment becomes so oppressive or this thing not conducive for their proper development, all these things shut down. Similarly, even in the human body, the genes are allowed to function normally depending on these things. But one very important thing every one of us have to keep it in mind is, for these genes to function properly, you have to maintain proper physiological environment. That is basically because for example, physical activity is more important for all the cells to be functionally correct, doing well. So is the case even for the genes. Okay, defective genes are, you cannot stop. But earlier, this thing, earlier, when there are defects, they can lead to some kind of a problem. So till very recently, where Nobel Prize was given for two women scientists, Jennifer Doudna and Emmanuel Charpentier, two women, they actually found out how to correct the defective genes. Gene editing, you must have all read in newspapers. Now we have the technology to edit the defective gene. Earlier, if the defective genes are inherited, nothing can be done. Only palliative thing. Of course, gene therapy and all that thing, but gene therapy was successful in some areas, but it was by and large was not successful. But now you can actually correct these defective genes. You may say, how can we actually correct these genes in the entire human beings where there are 100 trillion cells? But through genetic counseling, you can identify what is the risk factor from this male and female to have a defective child, because we'll go into the history of their parents, grandparents, great-grandparents. From them, you can identify what would be the probable defect as far as some genes are concerned. If there are that in the ancestors of any of these people, you will identify, then you will also look for the probability of those things. Then you can come to that conclusion, what is this thing? Then, but the couple are not satisfied. They are not happy by having 25% risk, 50% risk or this thing. They want to know for sure whether this unborn child is going to be perfectly normal or defective. And now that can also be tested through prenatal diagnosis. So, and this through prenatal diagnosis, you can identify whether this particular defect that you identified through genetic counseling, whether that really happens in this particular child or not. You can identify from this some amniotic fluid, from this amniotic fluid, some DNA cell, you can make DNA, and that DNA can be tested whether that DNA actually has this particular defect or not. If it does not contain, you can allow that couple to have a normal child. On the other hand, if a defect is found, you can advise this couple to go for abortion. So that is the thing. Now, even if defect is found, that can be corrected through this CRISPR-Cas gene editing technology. So that's one, right? Another very important thing, important thing is, because the basic genetic material is same from virus to human beings. So earlier, you must have all read about hybrid paddy, hybrid wheat, hybrid animals and all that thing. So in hybridization, you can have this kind of hybridization only within the species. Among cows, you can have, among each goats, all these things. But here, in the process of hybridization, no doubt you'll get some of the best qualities that are present in both the stocks, but along with that, some bad qualities may also come. There is no way to avoid the bad qualities when the process of hybridization, but that can be avoided here in genetic engineering. Genetic engineering is, you can take any gene from anywhere and introduce to any species. This is possible basically because the basic genes are constructed from the basic building blocks. Now you will find, so the possibility of genetic engineering is, you can grow apples on neem trees. What is the genome that is making the apples to produce apples? If some of the genes which are crucial in making them apples, they can be taken out and introduced into neem seed. When you actually introduce those genes into the neem seed, the neem tree grows and will give apples. This is the maximum possibility through genetic engineering. The only thing is, I'll give another example. The greatness, the enormous potential of genetic engineering. For example, now the climate, the environment. To give you one example, for example, oil slick. So clearing oil slick on the ocean is an enormous task. It takes long time and then a lot of money. Now you can create some bugs, some bacteria, which will grow on this oil. They proliferate. They don't cause any harm to the environment. They proliferate. Now, some of the scientists, for example, Craig Venter was also one of the groups which were involved in identifying the Human Genome Project. He is making some cells which will take all the carbon dioxide that is there in the environment and produce usable fuels, usable food things, because whatever that is being produced is basically because of the genes. So he is preparing some kind of cells. He has already made one artificial bacterium, and this is dividing and functioning well. So the one big this thing is, all these things are happening in nature. Scientists earlier used to physical methods, chemical methods, and these methods. When scientists started observing what is happening within these biological systems, and then when you can replicate these things, then it can also give the results. I'll just give one example. Normal fertilization, procreation, right? The spermatozoa fertilizes the egg and all that thing. Suppose there is some problem in this fertilization process. That is now can be done, the fertilization process per se, in the laboratory, in vitro fertilization. This likewise, all that is happening in the biological systems, each gene, how it is behaving, when these things were studied, they also can be repeated in the laboratory, and then that is this thing, right? Through genetic engineering, the greatest advantage is two things. We can produce all the foodstuffs that are needed for the ever-growing population by increasing the yield of these things, and also you can also produce the best animals, all those things, the best animals, not only for the production of not only meat and all that thing, but other enzymes, proteins, and all that thing. Above all, earlier, when there are genetic diseases, nothing can be done. Now you can produce human beings who can live healthily for 150 years. So the health of the humanity can be improved enormously, and the food stocks, you can amplify manyfold the yields of all kinds of foodstuffs. So these are the possibilities, and this genetic engineering is the greatest technology that will help humanity enormously in overcoming. Because what are those things? We have to be healthy and we have to live well, and these things can be achieved through genetic engineering. And if I want, I mean, I'll show some of these genetic engineering, right? So this the Human Genome Project. The enormity of this Human Genome Project, I'll explain. I told you that there are 100 trillion cells. If you take DNA from each cell, and each cell, I told you, 6 feet. And if you put it one end to one end, all the DNA made from all the 100 trillion cells, from Earth to Sun, you can go 6 times and come back 6 times. That kind of DNA is there in each individual. And the functioning of that individual, these genes are so precise, nothing can go wrong. But, right, another very important thing is, many of us are getting cancers, like human beings. And the genes responsible for the normal cells to become cancerous is only 10 to 15%. The other 80 to 85%, we are putting on ourselves for the genes not to function properly, rather to make these genes undergo some mutations which will transform the cell into cancerous cells. So there are certain things that are to be observed in each every human beings for the genes to function properly, and those things are like the మన వీటిలన్నీ చెప్పింది అనమాట అండి. ఏంటి? మితాహారం, ఎక్సర్సైజ్ మంచిగా ఉంటుంది. బికాస్ epigenetic factors, your epigenetic factors are doing lot of problem because of our anger, jealousy. All these things actually create some cortisol within the cell, which will inhibit the proper functioning of the cell. So right. So these are the things. And then another very important thing is 99.8% of the sequence of the genome is same exactly in every 2 individuals on this globe. But that 0.2% of the difference constitute out of 3.2 billion bases. That comes to 300 million. And 300 million is a big number. And those changes are the reason for every individual to be unique. They are unique and different. So, and that is basically because of the genome. Right. So now technology has improved. Earlier, the Human Genome Project, it used to cost $1 billion to identify the sequence of the DNA in one individual. Now it has come down $1,000 because the technology has improved so much. By spending $1,000, you will identify the sequence of all the individual, all the genes. This 3.2 billion bases can be identified with 1,000. And in future, even with maybe $100 or maybe like 10,000 rupees, we can have these things. When once we have that particular sequence, you can identify the strengths and weaknesses of each individual with regard to all kinds of genes that are this thing. Based on that, what kind of care one should take, what are the strengths and weaknesses of their genomes can be identified. For example, to give you some this thing, nowadays, because COVID, I'll explain based on this COVID. It seems in the process of human evolution that there at one stage there were one kind of human beings known as Neanderthals, and those Neanderthals then actually interbred with the Homo sapiens, and it seems one chunk of DNA from Neanderthals were inherited, passed on to these things. It seems 50% of the South Asian populations, South Asia is nothing but India. The other things are there, other countries are there, very small. The overwhelming majority of South Asia is India. So 30% of the Indian populations have inherited but that particular chunk of DNA which is present on chromosome 12. And if those people who have inherited this particular chunk of DNA, if they get COVID infection, there is every likelihood, in spite of all the best medical care, they die. They are dying. On the other hand, another 30% of the South Asian populations have inherited another chunk of DNA from Neanderthal people, and all of them are protected against this COVID. So even if they get COVID, this thing, it's like no more than a cold and all that thing. They'll have fever or body pains and all that thing for a week, and afterwards immediately they are out of woods. But on the other hand, those people who have inherited this particular chunk of DNA, are vulnerable, susceptible. In spite of the best medical care, they are dying. So once we have this sequence of DNA, we will identify what are the strengths and weaknesses. So ultimately, we have to have this genome sequences of each and every individual because of this computer IT and all that thing. Now, with a click of a button, you can identify the vulnerabilities, the strengths, how they have to be treated in case if they are actually got any kind of infection. What are the chances for heart disease? What are the chances to become diabetes? What are the chances for become somebody to get cancer? All these things can be identified through the basic human genome sequence. And within this human genome sequence, there are, I told you, this thing, exome sequence. They are actually involved in creating, the proteins and the enzymes. And we do not know what is the other thing as of now is doing. But once we have this thing, a lot of research is going on, thousands and thousands of scientists are involved in identifying what each chunk of genome is actually doing. Very soon, we will identify the functioning of each and every part of this human genome. Then we can, this thing. That is why I told you, because of this human genome, these things, and because of genetic engineering, you can make experiments in such a way you can ensure the normal, healthy life extended to 150 years. That's one. And the food production, food security can be enormously increased to unimaginable these things, because human population is growing, arable land is shrinking on the other hand, because of the construction of all these things and all that. Of course, we can bring some of those mountains and all that thing also into cultivation, but still there is a great potential to improve the yield of each and every crop and animals, and of course for the human security, this thing. And I'll stop here, and I'll be more than happy to answer any of these questions. I'll try my best to give if I know, I'll tell, or else most probably, as far as these things are concerned, I think I know something. Right. Thank you again,
so
once
much,
again,
sir,
to all of you for giving me this opportunity.
Thank you so much, sir. Thank you. Yeah. If you have any questions to be asked, please unmute yourself and ask Professor Paidaya, please. Thank you. Yeah, unmute yourself, James. You can unmute yourself.
Right. Yeah, please. I'll be happy. I mean, in fact, if you don't ask me questions, I feel bad.
Thank you so much, Professor Paidaya. We had a very comprehensive lecture, and so much knowledge we have learned. And though we know a little about genetics, but we understood more clearly that how much our body constitutes of these build blocks, these materials, and how much they affect our well-being from all our organs to our behavior patterns and to how we look like. And so all these are determined by genes. And so I understand, and these genes can also be very ancient, sir. These also.
Right, sir. Right. Very, very ancient. Because see, right. we share 30% of our genes with bacteria. Some of the genes which produce some of these sugar, all those things, they are there in bacteria, they are there in human beings. So across this evolutionary ladder, all these genes are preserved. There are some additions, deletions within this genome to make it more perfect. To give you one example, the dogs have a lot more genes to identify smell. Pigs have a lot more genes to identify n number of smells. As the evolution took place, some of those genes were lost. That is why we can identify only one range of smells, not as many variety of smells as the dog or the pig can identify. So, right, I think, yeah, this is the thing.
Understood.
Right, yes.
So basically, we can say that we are related to bacteria.
Of course, yes. I mean, as I told you, all these things are social constructs. The caste, the religion, the region, the nation, all these things are only social constructs, but the basic building material. But there is some kind of this thing, because people started observing endogamy among these castes. There is a possibility for some of the genes to get segregated, consolidated. But that does not mean they are entirely different from, they are not sharing from other things. There are, yes, because of hundreds of years of this endogamy, there is a possibility some genes like what is called genetic drift. They got actually drifted in that particular community to have more. That does not mean those genes are not there in other castes.
Yes, absolutely. Understood, sir. And one more question, sir. So, for example, if I'm curious to know that how much part of my DNA is from Neanderthal, or as you said, 30% of had were not being affected by COVID, 30% were getting affected, not affected, and 30% were getting affected. So where should I go?
Right.
Check.
Sir, right. I think once we have the genome sequence, we can identify. But getting that, because that is I think in near future, the government of India should have these things ready, so that in future, no matter what kind of pandemic or endemic or what kind of infection that comes, you can identify who are the people vulnerable, who are the people who can withstand this thing. But right now we can identify, but to have this genome sequence, you have to have, for example, CCM can identify these things, but it costs a lot of money. But still, it has not come to ordinary this thing. Very soon, because it's like cell phones, cell call. Earlier, it used to cost a lot. Now, this thing. Similarly, if the whole world is going for human genome sequence, then with 1,000 rupees, I think one can have the entire genome.
Right.
I think it's only in the near future. Yes, in maximum in a decade's time, we can have this genome sequence of each and every individual out of all this 7 billion or whatever people. And then we can easily identify. And that is the future, because the future medicine, again, we don't, another very important thing is, you can dispense with all this pharmaceutical chemical formulations. The future medicine will be oligonucleotides, peptides, small peptides, oligopeptides, because when there is a problem within the gene, it produces a wrong amino acid sequence. So what is missing? If that particular amino acids are produced, if you take that thing, that will correct the problem. So you can avoid, because chemical formulations, one reason I'll give. Nowadays, because of lot of kidney problems are basically because indiscriminate use of antibiotics and other medicines. Because whatever we put into the system ultimately have to be removed by the kidney. In the process, is getting affected.
Yes.
So, yeah, in future, you can avoid the complete chemical compositions and look for biological things, small peptides. When what is missing in the gene can be replaced with this kind of the small peptides in large numbers, and that will take care of these things without causing any problem to the system.
Technology.
People are talking about artificial human development. How is it possible?
Right. Even in the genetic engineering, when you can improve the yield, that example I gave, you can grow apples on neem trees.
Yeah.
So you have to identify what is the genetic mechanism that is making in an apple tree to produce apples. You don't have to take the entire genome of apple tree, but those particular that chunk which is crucial in making these apples, you take that thing in here and a neem tree is producing. So that kind of a thing can also be done in human beings, but scientists themselves put an embargo on doing these kind of this thing because again, I had to explain, because biological weapons, I told you that you can create a bug which will clear all the oil slick. Similarly, you can also create a bug which, if it infects, my flesh will come like bread, and each cell has this infection, and each cell is capable of infecting another person. So it goes in, and it's like a nuclear explosion, nuclear, what is called fission. It can infect every individual and make them vulnerable to die. That kind of possibility is there. Similarly, science is always neutral. You can use it for the best of the society. You can use it for the destruction of the society. Starting from prehistoric times, like fire. Fire was discovered or invented by Neanderthal man some thousands of years ago. Even today, without fire, we cannot live. Fire can destroy the whole thing. So is the case. Any scientific development is always neutral. How it is used for the benefit of the society or the destruction of the society depends on in whose hands this is put. That is the only difference. Yes, you can definitely design the best of possibilities, like the Greek heroes, this thing, and all the geniuses. So when you identify this is the chunk which determines the genius, this is the chunk which determines the physical strength, all these things can be put together and then put it into these things. Then that particular individual will have all the desired qualities, right? It can be used for the best, for the worst.
So there's a possibility.
Oh, yes,
possibility
yes,
that superhuman beings will be there in the future.
yes, sir. Yes.
Madam Rama.
Sir, really, we had a wonderful lecture session, sir, today. I came to know many things today from this lecture. I want to ask you, sir, regarding this cloning concept also.
Right.
Can you share?
Yes, ma'am. Cloning in the sense, see, so far, the dolly, the conventional wisdom says that only spermatozoa and egg participate in the procreation process, right? But that Ian Wilmot defied the conventional knowledge that only gametes participate in the procreation of the next generation. But he has not completely this thing. What he took was, he took a fertilized zygote in which the nucleus was removed. In this place, he put a nucleus of that cell which derived from the udder of this thing. That's a somatic cell. When once a somatic cell becomes, the cell becomes somatic, it actually discharges its function. It cannot actually take up the procreational function. But he saw to it through some electrical signals and all that thing. This nucleus of a somatic cell started behaving as if it is a fertilized zygote. But here, the zygote, the other part is the conventional part. But that's a great thing because he saw to it that the nucleus of an adult cell started behaving and giving instructions as if it is a gametic, the nucleus of a zygote. That itself is something. The next best thing is we have to make, forget about even this fertilized zygote, taking away the nucleus and all that thing. Just take an adult cell, somatic cell, and make the entire thing. Because when you make the nucleus to give instructions, you can also make the other cells to behave as if it is a zygote, the zygotic cell. But it has to be worked out. Possibility is there, but it has not been worked out so far.
Sir, like that only Dolly.
Yes, ma'am. Yes, ma'am. Because Dolly again signed, some people said Dolly died early, not because of this kind of cloning, but because of some other problems. Because from Dolly's cell, 4 more clonings were done. They are known as Debbie, Daisy. There are 4. All of them are doing well.
Okay.
The mother died because of some other problem, not because of cloning. Because earlier thing was the understanding is because somatic cell lived this long, it can live only the further part. It cannot start from the beginning again. That was the conventional way, I mean, this thing earlier in the explanation, but that was proved wrong. You can, when it started behaving as if it is a fertilizer zygote, it starts its life afresh. And that was proved in the cloning that was made from Dolly's cells.
That has been stopped there itself, no, sir? Not for cloning of human beings, not accepted by sources.
Right, sir. It is banned. Some Chinese people were trying somewhere because officially nobody can clone human things. If somebody, some scientist is found out that he is doing this kind of thing, he can be put behind bars.
Yes.
Because I mean, the problem is still we are not complete. We do not have complete information about the entire human genome. There are many missing links. Unless we have complete information about the entire human genome, it's not this thing good to start this kind of a thing. Because I gave the biological weapons. We do not know what kind of individual will arrive. In the absence of complete information about the entire genome, it's not good to go for this kind of a thing.
Yes, sir. Yes, sir. So are we using any artificial intelligence tools?
Of course, without artificial intelligence, we just cannot go anywhere. Because the greatest support for every experiment is coming through artificial intelligence. Because, see, I'll just give, by following our Facebook, those people will know much more about my behavior than I know myself about my behavior, because the type of choices I make, the type of interaction I think, the type of answers I give, all these things, based on that, the artificial intelligence has its own algorithms which will find out what kind of personality I have.
Yeah.
So, because see, this artificial intelligence reduces the drudgery, the burden, because but actual things have to be done by the scientists only. When
Yes.
all these things were identified and fed, because of some algorithms, it will give some conclusions. And there is every possibility for those conclusions to be right. There are, of course, some exceptions, even like a human error, because it all depends. It will not be even error, because if all the information is fed, that is known, because from unknown information, something may go wrong. But when the entire information is known, the algorithm will not go wrong. The artificial intelligence will not go wrong, because it can take all the billions and billions of cases into consideration, and in a second or in a few seconds, it can identify what is the problem. In that way, without computer, without our artificial intelligence, things will not go fast. I mean, yes, ma'am.
I used artificial intelligence tools to identify cardiac disorders in my research.
Right.
So similarly, genetical engineering, can we make use of that? Means I came to understand from your lecture session. Really, really wonderful, sir. Thank you so much for your patience, and you answered very well, sir. Glad
Thank
to
you.
see you. Glad to have your bright lecture today.
Thank you, ma'am. Thank you, ma'am. Thank you.
థ్యాంక్ యూ సార్. ప్రొఫెసర్ పద్దయ్య గారు సుమారుగా 1:30 సమయం పాటు మానవ జన్యు శాస్త్రం ఇటీవల పురోగతులు అనేటువంటి అంశం మీద సుదీర్ఘమైనటువంటి ఉపన్యాసం ఇవ్వడం జరిగింది. వారికి మా తెలంగాణ జనవేదిక తరఫున ప్రత్యేకమైనటువంటి ధన్యవాదాలు సార్. ఇప్పుడు మనోజ్ రెడ్డి గారు మనోజ్.
Really, thank you so much, sir, for giving your valuable time. Really, genetics, human genetics గురించి చాలా విషయాలు తెలియని విషయాలు చెప్తున్నారు సార్. Really, you are a gem in that department and in that subject. Yeah, really very happy to get introduction to you. And
I can see some of the features of Prashant in your face.
thank you, sir. Thank you. And we'll get in touch with you in much more meetings in coming days, sir.
Sir.
And thank you so much. Thank you again, sir.
Sir. It's my pleasure. Actually, talking to an elite audience is always it's a pleasure. Talking to students is always a pleasure, but talking to elite audience is much more happy and pleasure things. And thank you very much for giving me this opportunity, sir.
We're very privileged to have you here. Thank you so much.
It is really a privilege.
Right, sir.
థ్యాంక్ యూ సార్.
Right, sir.
తెలంగాణ జనవేదిక
అయ్యే సార్, అసలు హ్యూమానిటీ అంతా ఒకటే సార్.
ఓకే ఓకే సార్ అందుకే మేము తెలుసుకోవాలని అడిగే ఫస్ట్ అన్నాం సార్. మేము సోషల్ సైన్స్ వాళ్ళము ఈ జెనెటిక్స్ గురించి తెలుసుకోలేదు, మాకు తెలియాలనే మీ లెక్చర్ లో చాలా ఇంట్రెస్ట్ గా విన్నాం. చాలా విషయాలు మేము తెలుసుకున్నాం సార్. మీరు మా తెలంగాణ జనవేదిక కచ్చి కీలకోపన్యాసం ఇవ్వడం మా వేదికకు చాలా ఆనందంగా ఉంది. మా వేదిక తరఫున మీకు ప్రత్యేకమైనటువంటి ధన్యవాదాలు తెలియజేసుకుంటున్నాం సార్. అదేవిధంగా ఇవాళ కార్యక్రమంలో పాల్గొన్నటువంటి మనోజ్ రెడ్డి గారికి మరియు జేమ్స్ ప్రేమ్ కుమార్ గారికి, ఇతర మిత్రులందరికీ పేరు పేరున ధన్యవాదాలు తెలియజేసుకుంటూ ఈనాటి ఈ కార్యక్రమం ఇంతటితో ముగిస్తున్నాం. వచ్చే ఆదివారము మరొక అంశంతో కలుసుకుంటామని అందరి దగ్గర సెలవు తీసుకుంటూ అందరికీ మరొకసారి పేరు పేరున ధన్యవాదాలు. థ్యాంక్ యూ సార్. థ్యాంక్ యూ ఎవ్రీవన్. థ్యాంక్ యూ సార్.
Thank you so much, sir. Thank you so much. It was really
Thank you so much, sir.
a privilege to have you here. And we were really enlightened by your talk.
Thank you, sir. Thank you. Thank you
so
very much.
sir. Thank you so
Right,
much.
sir. Right. Okay, sir. I'll take leave of you.
Thank you, sir.
Right, sir. Right, sir.
Thank you, sir. Goodbye.
Thank you, Ramu.
Thank you.